Hello everyone, and welcome to this special edition of the Dieting Dietitian. Today, we are here with Dr. Mark Brudnak. Now, you have a very impressive resume. Can you go into your areas of expertise?
Sure. I'm traditionally trained as a molecular biologist slash glycobiologist.
Okay.
My first dissertation was on the investigation of a novel eukaryotic cloning and cellular engineering system.
Easy to remember. I got that, no problem.
Yeah, it's a mouthful.
Yeah.
Basically, simply put, what I did is I created a cell line that allowed you to clone in complex cells versus bacteria.
Okay.
But I had to use bacteria to do it. And when I say my background's in molecular biology is simply the use of enzymes to cut DNA. And you traditionally do it in bacteria. You cut the DNA and then you move it around with different enzymes. And so a lot of my background with enzymes and probiotics comes from the fact that you have to understand bacteria, you have to know how to grow them, you have to know how to use enzymes and the kinetics and the conditions under which they function properly. And so that's that aspect of it.
Now, can we go into enzymes? Can you give me a very simple definition of what an enzyme is, and if you want to go more into detail?
Absolutely. An enzyme, simply put, is something that allows a reaction to occur with less energy.
Okay.
And so they do that by bringing bonds close together, typically, and it allows new bonds to form. Old ones to break, new ones to form.
Yeah, I know that in my college levels, they always talk about like a lock and key kind of effect.
Exactly.
Okay.
Yep.
So.
And as the key goes in the lock, what it does, the key would be a substrate, something that the enzyme works on. The enzyme would be the lock, and the key goes in. The lock actually changes shape, which we call conformation. Okay. And then that allows it to break bonds in the substrate and create new products.
Okay. So like for food, if you have a big piece of apple, chew it and start breaking it down, and it's still in a big carbohydrate form. Those enzymes help with that unlocking to make it into smaller pieces. Is that a very...
That's exactly what they do.
Okay.
Yeah.
Okay. Very
well put.
When you look at enzymes for overall health, what are you focused on right now?
Well, traditionally the industry, the natural products industry, has revolved around food enzymes and the use of those to help digest food. However, I see it shifting. There's going to be a move as they become more available towards metabolic enzymes and detoxification enzymes and things like that.
Ooh, okay.
There are thousands of different types of enzymes that we make and have access to. But the problem is purifying them in high doses where you can supply them in large quantities.
In clinical doses. That that makes sense. Okay. You also own a company and you make, I guess for lack of better words, enzymes and probiotics.
Correct.
Okay. I know we talk about it for helping with our digestive, system. Are there any other benefits of probiotics?
There are a lot of benefits. As you mentioned, they help digest things. They can increase the immune response. Well, they can decrease the inflammatory response, which is huge. They keep bad bacteria like Salmonella, Listeria, E. coli, Campylobacter, things like that. Keep those at bay.
Now, not only bacteria, but it's also beneficial for issues involving yeast overgrowth. Is that correct?
Right. Yeah. And so when we talk about yeast overgrowth, it's usually Candida albicans.
Okay. Yes. Okay.
As opposed to Saccharomyces boulardii or something like that.
Okay. Good. Yeah. Thank you. Yeah.
Sure.
Again, I always think about Candida.
Yes.
The yeast. What is a prebiotic?
A prebiotic is the food that the probiotics eat. Something like INULIN or FOS, and there are others.
Okay. And then, so the probiotics need the prebiotic for nourishment.
Correct. And the other qualification on the prebiotic is it can't be used as a food by the pathogenic bacteria.
Interesting.
And that's, because otherwise, all of life basically, not all of it, but the vast majority of life utilizes glucose and that includes good and bad bacteria.
Okay.
So you could get the probiotics to grow significantly just by supplying them pure glucose. But the problem is solely E. coli and all the other bacteria.
Interesting.
Yeah.
So there's certain prebiotics that only feed good bacteria, not bad bacteria.
Correct. And that's part of the definition.
Something that I've been hearing a lot about recently is something called a postbiotic. Can you tell me about that?
Sure. The postbiotic is a new concept. When we were growing probiotics in bulk, what you do is it's called fermentation.
Okay, yeah.
You grow them in big containers, and then you separate them from the media, and then you dry the probiotics. Well, we take the media. When probiotics grow, what they do is they secrete things into the environment, and also they release them when they break. And so probiotics follow the standard growth curve of any bacteria, and it's hyperbolic. There's a lag phase, and then an exponential phase, and then S phase up here, and then they start to decline. But as they're growing, they're secreting things in the media. And what happens when they hit exponential phase is the media becomes conditioned with the things that the probiotics are releasing.
Okay.
And so the media becomes this super environment, if you will, for the growth of probiotics. And so we take that media and we dry it, and we found that when we add it back into, or with the freeze-dried probiotics, that metabolites, which we've trademarked as MAC-tabolites, have a prebiotic effect, even though they were post-growth of the bacteria.
One thing that I read about is something called short-chain fatty acids in the postbiotics.
Correct.
Now, I mean, what is that good for your digestive system?
Well, the short-chain fatty acids, things like butyrate, for instance, are the primary fuel source of colonocytes, the cells of the colon.
Oh, really?
So without those, and that's one of the big things that bifidobacteria do, because the bifidobacteria are found in the lower intestines, the colon, and lactobacilli, as a rule of thumb, are found in the upper GI, from the mouth on down to the small intestine.
So lactose in the little or the small intestines, bifidos in the big or the large intestines.
Correct.
Got it. Usually. Got it.
There's some crossover, but
you developed a very unique way to make sure the probiotics survive the stomach acid to get into the small and large intestines. Can you tell us about that?
Sure. It was 2 steps, actually. The first was the creation of what we call MBT, or MAC-track bypass technology. And what that is it's a complex marine polysaccharide that from brown algae.
From algae, okay.
Which Dr. Mitchell had mentioned, or calls a seaweed submarine. And what it does is it forms a secondary capsule in the stomach acid that keeps the inside of the capsule completely dry, like the inside of a submarine.
Okay.
And then that was the first step, was the creation of that, and that as the pH increases with the passage into the small intestine, it causes a gradual release of the contents of that secondary capsule, and which is what you want. So sort of, it's a gastric bypass system.
Okay. So pretty much you mix this seaweed with the probiotics, with that stomach acid, it forms like a barrier. So then the stomach acid is not killing all the probiotics, the good bugs that are in there.
Exactly.
Okay. So then it survives the stomach, it gets into your small intestine, and then it starts breaking apart. What happens after that?
Well, what happens after that is why we created the MAC-track 3D system, which is the second part of the whole system. So what MAC-track 3D is we utilize the MBT to get it past the stomach acid, the seaweed submarine. But then we realized that nobody, including ourselves, up until that point, was really paying attention to what happened after that point. And you still have the vast majority of the digestive system to contend with. And so right after passage from the stomach, the bacteria get hit with bile. And so you've got this hollow tube, which are the intestines, and you've got this capsule sitting somewhere inside that. And the conditions on this side that the bile comes in, the conditions on this side of the intestine are vastly different from the condition right where the bile enters.
Okay.
And we know that that affects bacteria. For instance, we found bacteria growing on the sulfur vents in the bottom of the sea. And the closer to it they are, the better they grow. And there's a lot of life that feeds off of them and that. And so the same sort of thing. You have varying conditions, and we call these microenvironments. Okay. So the microenvironment of that capsule is what you're really concerned about.
Okay.
So what are we concerned about? We're concerned about the acidity and the alkalinity. We're concerned about the biosalts as well. So what you have a vast fluctuation of pHs and acidity levels in there. So what we did is when the probiotics are in a state of stasis, they're sleeping.
Yeah, because you do a gentle freeze drying process. So it doesn't damage them, it just kind of puts them into like a hibernation.
Exactly.
Yeah,
And in fact, when you're growing the probiotics, you said a gentle process, and it's very important that it's done right, because you can put them in a centrifuge, and that centrifuge can spin fast or it can spin slow. If you spin fast, it's very hard on the bacteria.
Okay.
There are pressure forces.
Okay.
You can do it in a much slower manner, and they'll form essentially a pellet, a concentration of the bacteria that you then pour off the media, and then take that material and you freeze dry it.
Okay.
Freeze drying is important in the process of doing it, because you can form different sorts of isocrystals. Some which will damage the probiotics, and some which won't.
Okay.
So you can have really sharp ones, and you can have ones that aren't going to be piercing the walls. And there are things that are conditioners in the media that when you dry it, they help plug the pores, the nuclear pores of the cells. Yeah. So that water can't get in. Water is usually your enemy, but not always.
The Magtrack delivery system bypasses the stomach acid, gets into the intestines, and starts breaking up. You added in INULIN, which is a food source, and you added electrolytes.
Correct.
What's the benefit of doing that?
As our friend Dr. Mitchell has explained it before, it's if you're a hibernating bear, what are you going to need when you're done hibernating? Because that's basically what they're doing. They're hibernating. And they're going to need something to drink, they're going to need fluids, and they're going to need food. And so we basically provide a Gatorade and a cheeseburger and get them going right away. And so the Gatorade provides the electrolytes and sugars right there along with the inulin. And the inulin really is the food, but the electrolytes are done in such a way that it balances out the pH in that microenvironment. And so it's an isotonic solution right in that environment. And if you don't have that, if you have too high a salt concentration in that microenvironment, they will take in too much water and they'll explode. But if there's too little salt, then they'll crinkle up.
Shrink.
And the problem with that, it doesn't seem like a big deal to us, but it affects how the DNA functions. A high salt concentration causes DNA to change shapes. And so the DNA drives everything that a cell does. Everything.
So really you figured out a way to do the right amount of electrolytes so they didn't drink too much or too little fluids.
Yeah, exactly.
Perfect. But then you also went one step further because you figured out a way to condition the probiotics to be resistant to things that are in our everyday food and water supply that otherwise could damage them.
Correct. For instance, fluoride, what we did is we found that ours could grow through extremely high levels of fluoride, which is important because the levels that we tested are typical levels that would kill things like strep mutans, which is carries the cavities causing bacteria, and other lactobacilli, in fact. But ours were able to grow through high levels of it. And we also checked it for things like alcohol, because it's a commonly consumed thing in our society. And we also checked things like acetaldehyde. And they can grow through high levels of acetaldehyde. It's important because it's 30 times more toxic to human cells than alcohol is. And it's the natural breakdown product of alcohol. Alcohol breaks down to acetaldehyde and then to acetate.
So pretty much you helped these probiotics become resistant. So if someone drinks, fluorinated, chlorinated water, or they have a beer, those probiotics are more likely to survive and still give you the beneficial effects.
Exactly.
My last question for you, Dr. Brudnak, is you did an incredibly interesting dissertation. Can you tell us about what you did your dissertation on?
Sure. The second one that I did was on molecular rationale for enzyme therapy. And what that basically means is that since the 70s, there was a notion that humans only have a certain amount of enzyme producing potential.
So you mean like a finite amount? Like some people thought at one point that once you used all your proteins to break down protein, that would be it?
That's exactly what they thought.
Wow,
yeah.
okay.
And they thought that that's what happened as you got older, and it was a significant source of aging. And so, as I explained earlier, I'm classically trained as a molecular biologist, hardcore science. And so I looked at it with some skepticism.
Okay.
But I looked at it also from the standpoint of everything that we do at McWood. We have the perspective that the body wastes nothing. Nature wastes nothing. It doesn't. It does something with everything, even breakdown products. Nature will do it. A good case in point is there are bacteria that will even digest PCBs. And so nature doesn't waste anything. So what I did is I looked at the problem of enzyme potential and realized that perhaps what could be happening is over time the DNA that makes the enzymes could become mutated. And so I started digging into it, which would result in loss of enzyme ability to produce enzymes. And so I dug into it, and it turns out you need a couple things. First of all, you need to show that the DNA, which is normally supercoiled and tightly wrapped around proteins, histones, to protect it. Because when DNA is closed, it's protected. Radiation can't affect it. Chemicals can't really affect it. That's why DNA can persist for millions of years. Is that it's a highly stable molecule when it's packed. Packed, especially when it's packed and dried. So I knew that we would have to determine a mutation that increased mutations when you open it up. So I found references that supported that, and so I was pleasantly surprised. So I knew that that was part of the puzzle. So you can determine increased mutation and loss of function of not just enzymes but other genes as well as a result of frequency of opening and closing the DNA. So the longer it's open, the more exposed it is to chemicals and radiation, oxidation, things like that. And the mutations just change base pairs basically, and it doesn't function as well, and eventually it'll lose its function. But the other key to this was how would supplemental enzymes affect the DNA? Because you're taking them, how would they even get to the cells? So what I found is that there's actually what's called an enteropancreatic circulation and feedback mechanism. So I found references that supported the idea that when you take enzymes, they're actually absorbed. Not all of them, some get broken down, but some actually get absorbed intact, parts of them, and they make their way back to the pancreas that produces amylase, protease, and lipase, the big ones. It produces other ones, but those are the big digestive enzymes. And so I found supporting documentation that showed they got back there, in that when those enzymes got back to the pancreas decreased its own production of those enzymes. Now, in order to produce the enzymes, as I said, the pancreas has to use its cells. It has to open up the DNA and produce enzymes. But if you're taking exogenous enzymes, what's happening is it allowing the pancreas to essentially rest and not use its own cells and its own DNA to produce enzyme. So the body knows what you're eating, and it's not going to waste energy producing enzymes that it doesn't need.
When you eat a meal, is your body so intelligent that it goes, okay, you had a lot of protein, we're going to release more protease, but not as much amylase?
It does know to do that.
That's crazy.
Yeah.
Now, and it's not like if you did the paleo diet, and then all of a sudden the next day you started going, would it take a while for your body to adjust and start releasing more amylase, or does it know pretty much instantaneously?
It knows pretty quickly.
That's
I mean,
incredible.
it's got to get absorbed, and there has to be a signal of some sort transmitted from that food, either by the food itself or by metabolite, something that's been broken down to, that then feeds back. But the body works, almost every single system in the body works by feedback inhibition mechanism. So the more something you have, you either, well, I said inhibition, but it can also positively affect it. The more something you have, if it saves the body energy not to have to do something, the body will then shut it down. But if the body needs whatever it is that you have more of, then it'll produce whatever it has to utilize that. And so it knows.
That's awesome.
Yeah.
So really, like when you do exogenous or take in enzymes by whatever method, capsule, pill, it gives your pancreas time to rest. But if you forget to take your enzymes, it's not like you're in trouble. You're still going to break down that food because your body knows to do that.
Exactly.
Okay, that's awesome. So I've had that question throughout the years where people go, oh, well, I've heard that if you take enzymes and your body stops making them. I've never heard that.
Yeah.
So
well, it doesn't. Yeah, it downregulates it. But when they say, oh, your body stops making them, they think that's bad. No, the body will start making them as soon as you stop taking them.
So it really is beneficial not only for health, but like to give your pancreas that break that it needs.
Yeah, and so as I said, anytime you open up that DNA to produce something, because the DNA has to open, you make an RNA, you make from which you make the protein, which are your enzymes, that it endangers that DNA. And so there are a lot of mechanisms in the body that repair the DNA, but they aren't perfect. And that's what results in us aging. So anything you can do to help the body keep that DNA closed, the better off you are.
Awesome. And it seems like enzymes are incredibly important, because I know we eat a lot of food that used to have enzymes and that doesn't anymore.
Yeah, absolutely.
So now, if you eat a lot of processed food, could there be a possibility that's harder on your body to break down more things or more food?
Yeah, absolutely. You may not notice it short term, but your body will notice it long term. We didn't evolve eating the processed food that we eat today. We just didn't. Our bodies were out there hunter-gathering. That's how our bodies evolved.
Yeah. That's awesome.
Yeah.
That's good to know, because I love enzymes and I
I know
them.
you do.
Yeah,
I do too.
Yeah, it's just so much, because especially if you're doing bodybuilding, you want to make sure that if you're taking in protein or these nutrients, you're counting your macros. You want to make sure that you're breaking down that food and utilizing it for building muscle, for energy, for glycogen replacement. It's really important.
Absolutely.
And of course, probiotics go right into that, because you need to have a healthy digestion tract in order to then absorb nutrients. So to me, enzymes and probiotics go hand in hand.
Yep.
Is that the way you feel about it?
That's exactly how I feel, yeah. I take both of them regularly.
There's 3 things I take every day, without a doubt. It's a multi, probiotic, enzymes.
Right.
Most days I take a essential fatty acid too.
Yeah.
Perfect. Well, I really appreciate your time, brother. It's been good to see you.
My pleasure.
Thank you so much for joining us. Please subscribe to my YouTube channel. Please check out my Instagram at Dieting Dietitian. Please check out Dr. Brudnak's Instagram, Macwood Inc. Yeah, he does updates. You can reach him that way. He's an awesome guy. He's been my friend for over 20 years. So yeah, we'll hopefully do more interviews in the future whenever our paths cross again.
I'd love to.
It'd be awesome. Thank you, brother.
Thank you.
All right, take care, guys. Thank you.